Hello everyone, welcome back to the channel. Today we are going to discuss the continuing part which we have discussed in the last video, that was single cross, or sir you can say 2-point crossover and 3-point crossover. To that continuation of this genetic mapping, we are going to discuss a very short concept regarding what is interference and coincidence. Okay, so we are going to discuss with examples so that you get clear. So whenever in exam genetic questions or numericals are asked, it is asked on the basis of this interference and coincidence also. It is the questions may be to find the genetic map, that is to find the gene distance, gene order, as well as to find the interference and coincidence. So it is a combined question, combined set of questions. Okay, so we have already discussed how to calculate gene map, that is gene order and gene distance, as well as we can calculate how to double crossover, single crossover. We have already seen in the last part of the video. If you have not watched that part of the video, I do recommend to watch that, and then you can watch this video for the better clear. Let's start. So before that, let's understand what is interference. So interference is nothing but to interfere, means always the crossing over which takes place does not occur in a uniform manner. Means the crossing over, we have seen what is crossing over. Okay, during the meiosis or during the cell division, crossing over takes place, and as a result of which exchange of genetic material takes place, but it does not occur always in a uniform manner. Means one chiasma formation of one chromosome will always reduce the probability of formation of another chiasma of the adjacent chromosome. Means what? If one crossing over takes place, it will interfere with the crossing over of another chromosome. So what will happen as a result? Double crossover will be reduced. The probability or the number of double crossover will be reduced, because crossing over is a result of double crossover is a result of crossing over. Okay, if 2 times single crossover takes place, then double crossover will happen. But if the crossing over is only reduced, then the probability of forming the double crossover will also get reduced. Due to that reason only, they have certain points. See, crossing over does not occur uniformly along a cross chromosome, and formation of one chiasma formation reduces the probability of another chiasma formation and of the adjacent chromosome, which we have already discussed. This lack of independence is called as interference. What is independence? Independence means that normally both the chromosome will undergo crossing over, but here what is happening? One is interfering with the independence or the activity of another. Okay, one is interfering in the work of another. That's why it is called interference. Okay, due to which we can see, as a result of which what happened, the probability of double crossover get less as we have expected. Suppose we have expected that the double crossover number will be 15, but due to this interference the double crossover becomes 10. Okay, so what happens? This happens due to interference. Okay, now we will understand that how to calculate this interference. Before that, let's understand that what is coefficient of coincidence. Okay, so coefficient of coincidence is nothing but the measurement of interference. Okay, so to understand what is coefficient of coincidence, we must understand certain points. That is, what are the conditions of the coefficient of coincidence. So there is a very simple formula to calculate interference, that is 1 minus coefficient of coincidence. Okay, where if you find the coefficient of coincidence, then you can just 1 minus coefficient of coincidence is the interference. Okay, then what are the conditions that are required for the coefficient of coincidence? You can see these are the certain conditions which are highlighted here. If coefficient of coincidence is 0, if coefficient of coincidence 0 means 1 minus 0. 1 minus 0 is 1, so interference will be 1. As a result of which it will show complete interference, means no double crossover will be seen. Okay. Second, if the coefficient of coincidence value varies from 0 to 1, means in decimal value, then also we will see interference and double crossover will be seen, but in less amount. Probability will be less. If it is equal to 1, means if coefficient of coincidence is equal to 1, so 1 minus 1 will be equal to 0. So interference 0 means there will be no interference in the double crossover. So the double crossover will be seen as expected. So remember one thing, if the coefficient of coincidence is 0, interference is 1. If coefficient of coincidence is 1, then interference is 0. Just the opposite thing. And remember, if interference is 1 means it is complete, no double crossover will be seen. If the interference is 0, then only double crossover will be seen as we have expected. This is a simple thing. Next, what happened? Next we will understand how to calculate this coefficient of coincidence. It can be calculated by a simple formula, that is number of double crossover or number of observed double cross divided by the expected double cross. Means what? What you are observing means you conducted an experiment, you conducted a cross and you observed certain value. That is known as the double cross which you find from this example or from the phenotype is the observed double cross. And what is expected double cross, which we are expecting. And how to calculate this expected double cross is a very simple formula. First we need to calculate the frequency of the expected double cross. That is the frequency of single crossover in the first region into the frequency of single crossover in the second region. Okay. Then you can calculate the number of expected double cross by is equal to expected double cross over total number of progeny. First you need to find frequency of single crossover, frequency of single crossover 1 and single crossover 2. How we will calculate that we will see in the example. So you can get clear picture. Then after getting the expected double crossover frequency, you need to just multiply with the total number of progeny. And you will get the number of expected double crossover. You have already, the number of observed value will be already given in the question, and you need to only calculate the expected number. By the ratio of observed by expected double cross, you will find coefficient of coincidence. And once you find the coefficient of coincidence, you need to just subtract it from 1 and you will get the interference, which is very, very simple. Next question is, how can we calculate the single crossover frequency 1 and single crossover frequency 2? The formula is very easy. It's somewhat similar to what we have calculated in the gene distance or the recombinant frequency. So let's see by the help of an example. Okay. So here is the Drosophila female with a genotype that is capital A, capital B, small b and capital C, small c, which was crossed with a male which have a genotype of small a, small b, small b. Okay. This is the genotype of the male. This is the genotype of the female. After crossing over, we get this following progeny, which is of 1000 in number. You can see here, these are the progeny and these are the number. In exam, sometimes the parental, which one is parental, which one is single crossover will be given, but most of the cases only this phenotype or the progeny and the number will be given, and you need to recognize which one is, and you need to identify which one is parental, which is double, which is single, which we have already discussed in the previous part of the video. Once again I am repeating the trick, that is, first you need to find the highest number, which will be the parental type, and the lowest number is a double crossover, and here this double crossover is the observed double crossover. Okay. The formula which we have discussed, observed by the expected, here the double crossover which we are observing in this experiment is the observed double crossover. Okay. Expected crossover we need to find out. How we will find out? For that you need to find out the single crossover bit first. Then you need to, after parental and double crossover, you need to find out the gene order. Okay. And the trick of gene order is to compare the first parental with the first double crossover and second parental with the second double crossover. And by this you can clearly see that the change is between B and C. So the gene order will be ABC. I have already mentioned how to calculate gene order in detail with the example in the previous video. I recommend you to watch that video for the better understanding. Next, what you need to do after finding the gene order, you need to find the single crossover between A and B and single crossover between B and C. And the trick also I have mentioned in the previous video, that the number next highest to the parental type will be the single crossover 1, that is between A and B, and then it will be next highest number will be between B and C. And to cross check it, you need to cross over and compare the value. I have already discussed how to do that. Next, after finding all the type, whether which one is parental, which one is double cross, which one is single cross, which one is single 1 and 2, next you need to find the single crossover frequency between A and B. And the formula is similar to that of the how we were finding the map distance, but the only difference here is that we are not multiplying into 100. If you remember the formula of map distance, it was number of recombinant by total progeny into 100, but here we are not multiplying 100. We are only calculating the gene frequency, that is single crossover frequency between A and B. So what is single crossover frequency between A and B? That is 152, here 152 plus 148, plus you need to add the double crossover also, that is 27 plus 23 divided by 1000. So you get the value, that is 0.35. Next, single crossover frequency between B and C is 81 plus 89 plus 23 plus 27. Double crossover need to be added in both the cases. Divided by 1000, that is 1000 is the total number of progeny which is already given in the question. If it is not given in the question, then you need to sum it on and you will get the answer. Okay. So 1000 that is equal to 0.2. So after finding the single crossover frequency between A and B and C, you need to find the expected double crossover frequency. And the formula for finding the expected double crossover frequency, which we have already discussed here, is single crossover in first region into frequency of single crossover in the second region. So we have already calculated here, that is single crossover frequency in first region is 0.35 into single crossover frequency in the second region is 0.22. After multiplying, you get the result, that is 0.77. Here only you have calculated expected double crossover frequency. Number you have not calculated. So how you will calculate the number? By this formula which we have discussed already, number of expected double crossover is equal to expected double crossover, okay, expected double crossover into total number of progeny. So what is the expected double crossover here? Here it is 0.077, okay, into total number of progeny is 1000. So you get the expected number, that is 77. Okay. And now what you need to do, you need to add the observed double crossover. Here the observed double crossover is 23 and 27. So after adding, you get the 50, which is the observed double crossover. Now you need to simply put the formula of coefficient of coincidence, which is observed double crossover by expected crossover. So here the observed crossover is 50 divided by expected crossover is 77, which we have got. Now you got the coefficient of coincidence. By this you easily can calculate the interference, that is 1 minus 0.675, because the interference formula is 1 minus coefficient of coincidence. So you get 0.35. So by this example you might be clear that how you can calculate from the sample, you can calculate the coefficient of coincidence, interference, gene distance, single crossover frequency, double crossover frequency, and all that. So in exams it will be asked in a combined manner. It will be asked to find the gene distance also, interference also, coincidence also. It will asked in a combined manner. So the previous two video which we have discussed, in exam the numericals will be asked in the combined manner of both. Sometimes it is asked individually, but mostly if you see competitive exams question, it will be asked in the combined manner. So you need to be clear with both the concepts, which is more or less the formula is similar, but in certain modification is there. So this is all for today. Hope you got what I have taught you. This is all about interference and coincidence, And if you have any doubt, you can ask in the comment section. I do recommend you to watch the previous videos of genetics so that you get clear idea what we have talking about. If you are new to the channel, do subscribe, like and share among your friends. So that's all for today. Thank you and have a nice day.